Hello my dear students, welcome to another session of class in ICSE Biology. In my previous video, we discussed about factors that affects the photosynthesis and also adaptation of plant for photosynthesis. In this session, we will be learning about some of the experiments on photosynthesis. Let's begin with the first experiment. The aim of the experiment is to test a leaf for starch. Materials required are a test tube, water bottle, Bunsen burner, a beaker, tripod stand, iodine solution, petri dish, and leaf. The first step of the experiment is to discharge the leaf. How can we discharge the leaf? We can keep the plant in dark place for nearly 24 to 48 hours, that is 1 or 2 days, so that whatever the starch is present inside the plant that will be utilized by the plant when there is no photosynthesis is taking place. In this way, we can remove the starch, whatever is present in the plant, and that is called destarching. So this is the first step what we have to do in the experiment. First, we have to destarch the leaf. Then second step, we need to take that leaf and put it in the beaker containing water and allow it to boil. Why we have to boil the leaf? So that the cells will be killed. The cells which are living, that should be killed before conducting the experiment. That is why first we need to boil the leaf in the water. Then we need to take that leaf and put it in the test tube which is having the methylated spirit or alcohol. So for few minutes we need to boil in the test tube which is having the alcohol. Why are we taking the alcohol in a separate test tube? Why can't we directly add the alcohol to the beaker and directly why can't we boil it? Find out the answer and comment in the comment box. After boiling the leaf in the test tube containing alcohol, the leaf will lose its color, that is chlorophyll. The green color which was there for the leaf, that will be lost when it is boiled in the alcohol. So now the leaf is not having the chlorophyll. Why are we removing the chlorophyll? So that when we conduct the iodine test, it should be clearly visible. So after that we need to take a beaker which is having the cold water and we need to add the leaf into the beaker. We are doing this to make the leaf soft. After boiling it in the alcohol, the leaf will become brittle. It means which can be easily broken. To avoid that breaking, we need to put it again in the water to make it soft. The last step is the iodine test to check the presence of starch. To do this, we need 2 petri dish and we need 2 leaves along with iodine solution. So what we are doing here, we are taking 2 leaves, one which is destarched, another one is normal leaf. Both has to undergo the steps what I showed before. The 3 steps what we followed, the same step we should follow for the both the leaf, but one leaf is destarched leaf and the other one is the normal leaf. After adding the iodine, one of the leaf will turn into blue-black color, the other one will remain brownish color. Why? Because the iodine solution color is brown, so the leaf will be brownish in color, the other one will turn blue-black. The leaf which turns blue-black, that proves that the leaf is having the starch, because the iodine solution has the capacity to turn the starch into dark color. So that proves that the leaf which is turning into blue black, that leaf is having the starch. The other one which is not turning into blue black, that will be the leaf which is not having the starch. If we conduct this iodine test by keeping the white background, we can see clearly the changes which is taking place in the leaf. One will turn into blue-black, the other one will remain as it is, the brownish color. In this way, we can prove the presence of starch in the leaf. The next experiment is to show that carbon dioxide is necessary for photosynthesis. The material required to conduct these experiments are a glass bottle with cork, a potted plant, iodine solution, potassium hydroxide flake, and a petri dish. To begin with the experiment, first we need to destarch the plant. You know what is the meaning of destarch. Already I have explained you. So first we need to destarch the plant. Then we need to perform the experiment. We need to attach the bottle to one of the leaf of the plant in such a way that half of the leaf is inside the bottle and the remaining leaf is outside. We need to fix up the cork by splitting the cork into two halves. We can also apply the Vaseline to make it airtight. The bottle should contain potassium hydroxide. Why? Because this potassium hydroxide has a capacity to absorb the carbon dioxide whatever is present in the bottle, so that the bottle is now free from carbon dioxide. There is no presence of carbon dioxide inside the bottle. Allow this setup for few hours and then take the leaf and test it for presence of starch. Same way, you know how to perform the iodine test. We need to take iodine solution and keep the white background. Place the leaf on a petri dish. Then add iodine solution. After adding the iodine solution, the portion which was outside that will turn blue-black. That means that has performed the photosynthesis. The leaf which was outside, which was getting the carbon dioxide, that will turn to blue-black color. That shows that the leaf has performed the photosynthesis and there is a presence of starch. But the part of the leaf which was inside the bottle, since there was no availability of carbon dioxide, that couldn't perform the photosynthesis process and it will remain as it is, normal color. This proves that carbon dioxide is necessary for the photosynthesis process to take place. Next, experiment number 3 to show that chlorophyll is necessary for photosynthesis. The following materials are required to perform the experiment: petri dish, variegated leaf. Let me tell you what is the meaning of variegated leaf. Here in this leaf you can see different colors. Here you can see the green color, this part is whitish in color, this part pinkish color, and this is somewhat a dark pink color. So like this, if the leaf is having different color other than the green, then it is called variegated leaf. So we need to select a leaf which is having the different color. So it means the leaf which we are taking, that should be the variegated leaf. A marker and paper and iodine solution. First we have to mark the outline of the green portion and the non-green portion on the paper with the help of marker, so that we will get the clarity for the portion which was green and the portion which is not in greenish in color. Then next we have to perform the iodine test. Same way, we need to take the iodine solution and we need to place the leaf in the petri dish which is having the white background. After some time we can see that the portion which was green in color, that will turn into blue black, and the portion which was having the other color than the green, that will be having brown color. It means it will not turn the color into blue black. This clearly shows that the portion which was having the green color, that portion had chlorophyll, and that will turn into blue black because in that portion the photosynthesis process will take place. Because of the photosynthesis process, starch will be present in that place and the iodine solution will turn the starch into blue black color. But the other portion which was non-green, that will not turn into blue black because there is no starch present in that place. This proves that the chlorophyll is necessary for the photosynthesis process. Next, experiment number 4. The aim of the experiment is to show that oxygen is produced during photosynthesis. The materials required are beaker, test tube, funnel, water plant. Here I have taken hydrilla plant and a water bottle. We need to fill the beaker with water, then place the hydrilla plant over that, place the funnel in this way. And then next we need to place the test tube over the funnel. So this is how you need to keep the setup for few hours. After few hours, we can see that in the test tube, the water level will come down because of the accumulation of gas. What is that gas? That gas will be the oxygen gas which was released by the hydrilla plant. This hydrilla plant will perform the photosynthesis process and it will release the oxygen, and that oxygen will go and collect in the test tube at the top and it will push the water down. So after some time we can see clearly the accumulation of oxygen gas in the test tube. How can we prove that it is oxygen gas which is accumulated in the test tube? We can prove that just by taking out the test tube, by closing the mouth of the test tube and releasing the mouth of the test tube in front of the burning splinter. When we release the test tube, the whatever the oxygen gas is there, that will come out, and because of that reason the splinter will burn little bit with high flame, and that proves that it is a oxygen which was present inside the test tube. Next experiment is to show that sunlight is necessary for photosynthesis. The following materials are required to conduct the experiment: a black paper, a scissor, petri dish, marker, iodine solution, and de-starched leaf. We need to cover the leaf with a black paper as it is shown in the picture. So in this way we need to mark either star shape or any other shape, or we can completely cover the leaf, one of the part of the leaf, with a black paper and allow it for few hours to perform the photosynthesis process. Then after that we need to test the leaf for the presence of starch. When we test the leaf for the presence of starch, we can see that the portion which was exposed to the sunlight, that portion will turn into blue-black. The other portion will remain brownish in color. Why? Because the portion which was exposed to the sunlight will perform the photosynthesis process, and in that place starch will be present, so it will turn into blue-black. But the other portion which was not exposed to the sunlight, that cannot perform the photosynthesis process and it will remain brownish in color. So in this way we can prove that sunlight is necessary for the process of photosynthesis. These are some of the experiments which proves that how the raw materials are necessary for the photosynthesis process, and also it proves that what will be the product of the photosynthesis. Hope it was simple and easy to understand. Thank you.